This lecture and the coming ones are a continuation of what Dr. Qusay has started with you last week, which are the antibacterials. But what we'll be covering are those antibacterials which are either isolated from nature, semi-synthesized from natural precursors, or even synthesized based on a natural scaffold. These antibiotic groups will be subclassified based on their mechanism of action. The first class that we will be talking about, which constitutes the majority of them, is the inhibitors of cell wall synthesis. Before we start talking about the structure-activity relationship and the various members of this class, we need to look at the structure of the target, which is the cell wall, and the variations between the gram-negative and gram-positive bacteria. This introductory information will be helpful in comprehending the chemistry behind the mechanism of action and the spectrum of activity. The cell wall, which is absent in mammalian cells, is part of the cell envelope that surrounds a cell membrane. The various functions of the cell wall are vital to the survival of bacteria. In summary, it's an incredibly complex superstructure that can be envisioned as a three-dimensional fabric that encases or surrounds bacterial cell, giving it its shape and resistance to internal osmotic pressure. Even though we're talking about the same naming of the structure for all kinds of bacteria, yet the composition of the cell wall varies among bacteria. These variations are the reason why bacteria stains differently with a gram stain, and they've used the gram stain to subclassify bacteria into many groups. Yet the two major groups that we will be talking about are gram-positive bacteria and gram-negative bacteria. The difference seen between a gram-positive and a gram-negative bacteria under a microscope is the relative thickness of the cell wall. That of a gram-positive bacteria is around 30 nanomolar, while the thinner cell wall in a gram-negative bacteria is around 10 nanomolar. Structurally, the main part that is responsible of the cell wall thickness variation is the peptidoglycan layer that provides the structural framework of the cell wall. The cell wall of a gram-positive bacteria is composed of many other structures alongside the peptidoglycan. Yet it's the peptidoglycan polymer that is of interest to us, since the enzymes responsible for its synthesis are the targets of the beta-lactams. These two figures are a 2D and a 3D representation of peptidoglycan. As the name indicates, peptidoglycan means that this polymer is composed of two parts, a peptide and a glycan. The glycan, or the sugar part, is a polymer that is composed of alternating units of NAJ and NAM. Those two sugars are bound together via a 1,4 glycosidic bond, which means that the glycosidic bond links carbon number 4 in NAM with carbon number 1 in NAJ, and beta indicates the stereochemistry of the linkage. Bound to the NAMs are polypeptides, which we'll refer to them as stem peptides. Stem peptides are composed of alanine, glutamic acid, lysine, and 2 alanines. Each stem peptide is linked via bridges to 2 other stem peptides. In many bacterial strains, the bridge is a pentaglycine. The amino acids bound to the bridges are lysine and the penultimate alanine. That process of such linkage is called the cross-linking, and during the cross-linking, which is basically formation of an amide bond, the terminal alanine is eliminated. The D letter that is seen next to some of the amino acids stands for the configuration of that amino acid. The configuration is opposite to the L configuration seen in natural amino acids, thus protecting the peptidoglycan in bacteria from being hydrolyzed by peptidases, which are stereospecific for the L configuration. The monomers of the peptidoglycan polymer are synthesized intracellularly, then they are exported extracellularly, where transglycosylation and transpeptidation occurs. Transpeptidation is the cross-linking that was explained previously. This process is catalyzed by enzymes, and the different isoforms of the enzymes responsible for the cross-linking are called penicillin binding proteins, which are the targets for the beta-lactams. The process of transpeptidation happening extracellularly requires that penicillin binding proteins be membrane bound, which renders them vulnerable to the effects of those beta-lactams. And once inhibited, the cross-linking would be inhibited. This will disturb the structure of the peptidoglycan, and for sure, this will affect the growth and repair of the cell wall, leading eventually to cell lysis. As for the thinner cell wall in a gram-negative bacteria, the peptidoglycan layer is much thinner than that found in a gram-positive bacteria, and they have an additional outer membrane, which makes it more difficult for antibiotics to penetrate gram-negative bacteria. The only part in the outer membrane that we are interested in are the porins. They control the passage of small molecules to the periplasmic space depending on their size, shape, and water solubility. This will be addressed again when we talk about the spectrum of activity and how it was extended via increasing penicillin's water solubility and was explained by the involvement of porins. The largest antibiotic drug class that we will be covering this week and the next weeks are the beta-lactams. Lactam stands for a cyclic amide, and beta is to designate which carbon is attached or covalently bound to the nitrogen. In any carbonyl containing compound, the carbon that is adjacent to the carbonyl carbon is designated alpha, the second one beta, the third one gamma, and so on. Thus, when the covalent bond is between the beta carbon and the nitrogen, that's when the lactam is called a beta-lactam. A more contemporary name for a beta-lactam is azitidinone. Azitidinone is derived from azitidine, which refers to the four-membered ring with nitrogen being one of its atoms. The different subclasses of the beta-lactams that we will be covering this semester include penicillins, cephalosporins, beta-lactamase inhibitors, carbapenems, and monobactams. These structures represent the various rings in beta-lactam subclasses. In most of them, there is a ring that is fused with the beta-lactam. The one that doesn't have a fused ring with the beta-lactam is the monobactam. Pinam is the nucleus of penicillins, and cepheme is the nucleus of cephalosporins. As for penem and carbapenem, they have a double bond between carbons 2 and 3, thus substituting the A with an E to account for the alkene functionality. The carba prefix in carbapenem illustrates the substitution of the sulfur heteroatom in penem with a carbon. As for the numbering, nitrogen, no matter what the subclass is designated number 1. In the case of monobactam, the beta carbon is assigned number 2. Yet in the case of the other fused ring systems, the carbon in the fused ring that is adjacent to the nitrogen is the one that is assigned number 2. The first beta-lactam antibiotic subclass that we will be covering is penicillins. The pharmacophoric ring in penicillins is the pinam, which is relative to a mono-beta-lactam, has a fused thiazolidine ring. Though an azitidine ring is not planar, mono-beta-lactam rings are planar. Planarity is favored so that the carbonyl atoms and the nitrogen, which are all sp2 hybridized, will have their p orbitals parallel, thus maximizing efficient orbital overlap and electron delocalization. Like acyclic amides, the nitrogen in mono-beta-lactams donates electrons via resonance to the carbonyl carbon, weakening its electrophilicity and consequently lowering its reactivity toward the nucleophiles. The pinam ring, on the other hand, adopts a V shape, where the nitrogen and the atoms surrounding it do not have a planar geometry. The hybridization of this nitrogen is not sp2, and the lone pair of electrons are present in an orbital that is not perfectly parallel with the p orbitals of the carbonyl pi bond, and this for sure disrupts the resonance, lowering to a great extent the electron donation from this nitrogen to the carbonyl carbon, making it more electrophilic and consequently more sensitive or more reactive towards the nucleophile. Also, the localization of electrons on the nitrogen renders the nitrogen more basic than that in acyclic amides and mono-beta-lactams. Another way through which we can explain the difference in electrophilicity between the carbonyl carbon in a penam and a monobactam or acyclic amide is to investigate the structure resulting when resonating the lone pair of electrons on the nitrogen with the carbonyl. The resulting structure has high bond angle and ring strains, thus it's highly unlikely to form. I believe you are all familiar with the well-known story of Fleming's discovery of penicillins. His accidental discovery of penicillins has advanced our ability to fight bacterial infections, saving millions of lives. It's a great example of how chance discoveries favors prepared minds that are always eager to search for answers. I just thought it would be nice to show you a picture of the first page of the paper, which was published by Fleming in 1929, in which he explains his finding. He included in that paper the original image of the culture plate from which he observed absence of bacterial growth surrounding a penicillium colony. Penicillium species biosynthesizes penicillin G, the first naturally isolated penicillin, using 3 amino acid precursors, which are cysteine, valine, and aminoadipic acid, giving rise to isopenicillinic acid. The benzyl side chain in penicillin G, highlighted in red, originates from phenylacetic acid that is added to the growth media of fungi. As a last step, it substitutes the amide side chain in isopenicillinic acid with the activated form of phenylacetic acid, giving rise to penicillin G. When substituting phenylacetic acid with other carboxylic acids, fungi produces other analogues instead. For instance, adding phenoxyacetic acid would result in production of penicillin V. 6-aminopenicillinic acid represents the pharmacophore of all penicillins. This precursor has a weak antibacterial activity. Some strains of fungi do produce 6-aminopenicillinic acid as it is, yet it can also be obtained via hydrolysis of penicillin G, penicillin V, or even isopenicillinic acid via acylases. When having the precursor in hand, this allowed the medicinal chemists to vary the side chain and produce a wide range of semisynthetic penicillins for the purpose of enhancing their drug-like properties such as oral activity, spectrum of activity, and stability against the beta-lactamases. All penicillins, including penicillin G, have an ionizable carboxylic acid which allows for salt formation. The nature of the salt, though, in terms of water solubility, will depend on the nature of the counter ion. If the counter ion is sodium, potassium, calcium, for instance, these salts would have higher water solubility relative to the free acid, as seen in the graph. Yet, if the counter ion is lipophilic, such as in the case of benzothane or procane, salts of this kind would have a lower water solubility relative to the free acid.